everybody. It's again pleasure to attend as every week the lectures by various colleagues. And today Dr. Prabavathy will be speaking about soil flora, particularly rhizosphere. Rhizosphere means, for many who don't know what is rhizosphere, it is primarily the root zone, soil and root zone, which has a lot of activity, particularly the bacterial and soil activity. That is called as rhizosphere. Dr. Prabavathy will be speaking to us on ecological hotspot for plant microbe interaction, soil interaction, and nutrient cycling. As you know, soil health card and soil has become increasingly important nowadays for increasing productivity, more particularly for enhancing water holding capacity of the soils. In this context, this study assumes very high importance. And you have heard Dr. Lal speaking in our Foundation Day lecture about how important it is and how many scientists, although they have not much come to the fore, maybe one of them is Professor Lal, the soil microbial flora and particularly soil studies are so critical. And today, Dr. Prabavathy will be speaking to us on this soil microbe interaction, particularly at the rhizosphere, which means the place where the roots absorb water and nutrients. That's how we define rhizosphere. Dr. Prabavathy finished her PhD degree in botany from University of Madras, and of course specialized in microbiology. And since then she has been working extensively on the mangroves and also interaction of soil biota in the mangroves. And without much ado, I request Dr. Prabavathy to speak about her very interesting. Although I have heard a couple of times, but I think it is very good for all of us to speak about. This is the area where not many of us will visualize what happens, like biotechnology and soil microflora. It is something highly imaginative and also brings out lots of very good stories. So I call upon Dr. Prabavathy.
Okay. Good morning. Good morning, Dr. Murli, for this introduction about the rhizosphere. And it's my pleasure to speak to the staff of MSSRF for the first time. I think I'm presenting our research work or whatever findings which is happening in the laboratory. Usually it is a PhD Viva OC of our students who present our work. But now I think this is the first time I'm speaking to the audience of or the staff of MSSRF. I'll try to be as simplified as possible so that you understand what I try to communicate. So with this, so today's topic, I would be focusing on the rhizosphere, as Dr. Murli had given a brief introduction. As he said, it is an imaginative region of the plant where interaction happens between the soil, the plant, and the microbes. It's a tripartite interaction happening, and it's a very fragile region and lot of activities happening which are interdependent on each other. So as he said, whoever speaks about soil, we naturally have to refer to Professor Ratan Lal, who has done a lot of work related to the physicochemical properties as well as to the soil organic or carbon content, exploring a lot into that area. But as he rightly pointed out during his talk in MSSRF seminar, that a lot of research has to go into the rhizosphere to really understand what is happening in the rhizosphere and how this interaction contributes to climate change. So during the World Food Prize talk, he had highlighted, to eliminate hunger, we must also make sure that the food consumed is healthy. And this is where the concept of healthy soil, plants, people, and the environment is a one and indivisible concept. So for the future generation, it is very important that soil resources must be protected, preserved, restored, and enhanced. This is where the future of the humanity lies. So these words speak about the importance of maintaining a healthy soil. So next, I just got this picture, which was from the FAO website, which clearly indicates how healthy soil is very essential for healthy life. It's a very, very complex process where it contributes not only to plant health, but also to human health, and a lot of biogeochemical cycles happening, the carbon cycle, nitrogen cycle, whatever cycles you call, and also which contributes to climate change happening. So soil management or maintaining a very healthy soil is directly linked to healthy human being. There again, it states that if you have a unhealthy soil, it naturally leads to unhealthy human beings. Next. So just to give a brief about the ecosystem services rendered by the soil, it provides food, fiber, and fuel, and it is a medium for carbon sequestration, where we are talking a lot about the necessity for sequestering carbon not into the soil. And also we know the ocean is a very big resource for carbon sequestration. And all these agriculture activities, is leading to disturbance in the carbon cycle and leads to the release of carbon, which is sequestered in the soil. And also it is a medium where water purification and happens, and whatever contaminants are dumped into the soil is recycled back by the soil organisms which are present there. As I said, it regulates climate, it contributes to nutrient cycling, and also it is a habitat of organisms. And also, apart from that, the physical processes like flood regulation, and it is the foundation for human infrastructure, and also a provision for construction material, and finally, it contributes to the cultural heritage of human being. Next. So with that brief background, all of us know that the Sustainable Development Goals, there are a few goals which directly speak about the soil sustainability and maintaining the soil functions and ecosystem services of the soil, where SDG 3 directly contributes to the agriculture soil. So I'm not going deep into by reading it out. Next. So I think I have already explained to you, so I just skip this slide, and just to give you a brief about how different the soils you can get. So it all depends upon the physicochemical properties of a soil, the ecological regions where they are present, and also, yes, based on the biological properties, you find the soil texture or the classification of the soil is very, very vast. And though there are a lot of reports, or the earlier decades, lot of research has been happening on looking into the physicochemical properties of the soil, but the current decade or the past one decade, we are concentrating more on looking into the biological properties of the soil, because now we understanding the biological property is very, very essential for maintaining the soil ecosystem or maintaining a healthy soil. Next. So as I said, there is a tripartite interaction in the rhizosphere region, where you find the plant, microbe, and the soil closely interact in the rhizosphere region. There are many processes which are happening directly or indirectly. For example, if you take an agriculture crop, the crop secretes certain exudates, which decides what group of organisms have to be present there. And then it also depends upon the type of soil which is present there, whether it is an acidic soil or alkaline soil. All this determines what has to be there and how it enhances the plant health or the yield of an agriculture crop. Next. So this, I just gave you a brief about the soil, and now coming into the soil microbiome. Just imagine the number of organisms which are present in 1 gram of soil. If you just take 1 gram of soil, you will see up to 10 billion cells of bacteria are present in just 1 billion of soil. Apart, and also fungi, if you take, it accounts to nearly 10 to power of 5 CFU units, and the actinobacteria nearly 10 to the power of 7. In addition to the protozoa, the nematodes, which are present there, and also the unculturable archaea, which are also present there. Here, if you see, understanding the soil biological properties, we have just explored only up less than 10% of the microbial population of the soil, but there's still 90% to be explored. But with the available current molecular techniques, we are still able to understand who are present there first. So it is very important to understand who is there and what is their function, how they contribute to the soil functioning. So, I think all of us would have experienced this. When there is a rainfall, you get a smell from the soil. This is nothing but it is just due to the actinobacteria which is present there, which releases a chemical substance called as geosmin, which gives that smell to the soil. So just imagine how much these microorganisms contribute to the functions of the soil. And apart from that, apart from contributing to the biogeochemical cycling of all the macronutrients and micronutrients, whatever material we dump into the soil, whether organic, inorganic, everything is recycled back into the elemental forms only by the microflora which is present there. So it is essential that we are very conscious that we do not alter this soil medium and put pressure on the soil microflora, because the soil microflora has a dual role in climate change. So it contributes as well as it is affected. So contributing to climate change would depend upon what material goes into the soil. So how it maintains a balance between the release of greenhouse gases and sequestration of carbon dioxide from the atmosphere. So just to show you the rhizosphere region. So when you see this picture, so our research focus mainly focuses only on that fragile region where the root zones are in contact with the soil. So you have two type of root zones broadly to classify, where you have a very good association of free living organisms around the root zone, and also a close association like the rhizobacteria. So my talk will be focusing only on the bacterial association, though you have the fungi, the arbuscula mycorrhizal fungi associated with the plant root rhizosphere. Our research focus is mainly only on the bacteria which are associated with this region of the root. So if you take, as I said, the root zone, it is a region of very close interaction where it harbors bacteria, actinomycetes, fungi, nematodes. All these organisms are present there. And this actually, the rhizosome bacteria is called as a second genome of a plant, because its influence on the bacterial genome or functions of a plant is highly influenced by the bacteria which is present in the root zone. So this doesn't mean that bacteria are present only in the root zone. It is present in the entire plant. You take the phyllosphere, the leaf, you have a separate group of organisms present there. When you look into the stem region or into the internal organs, you have the endophytes which are present there. But our research focus is concentrating only on the root zone, where we are looking into microbes which have specific functions like nitrogen fixation, phosphate solubilization, potash mobilization, and also micronutrient mobilization, which helps in nutrient cycling or makes the available nutrient in the soil or which are present in an unavailable form in a form that is available to the plant by secreting certain organic acids. So that is also a very complex procedure happening there. Apart from that, these organisms also help to maintain the health of the plant by producing a lot of secondary metabolites, which keeps away the pathogenic organisms away from the root zone, so that the plants do not get exposed to larger inoculum of plant pathogen and become susceptible to disease. So if you just look at the biological nitrogen fixation happening, so I've just given a figure there that the biological nitrogen fixation accounts to nearly 333 kgs of N per hectare, average to around 100 kgs. So this gives an indication that if we adapt strategies like using this biological material for nitrogen fixation by promoting efficient rhizobia or efficient associated microorganisms, we can really cut down on the nitrogen use in the agriculture crops at least by 50%. So next. So just to show what are the functions the plant and the bacteria in the rhizosphere has on the plant, you see it actually enhances nutrient cycling in terms of nitrogen fixation or phosphate solubilization, etc. And then you find it produces a lot of antibiotics, which keeps away the pathogenic organism away. It induces systemic resistance in the plant, so the plant is not affected by pathogenic organisms, and also elucidates certain induced systemic tolerance to drought, and so that the plant are able to withstand abiotic stress also. So all these mechanisms have been very clearly proven by a lot of pathways, which I won't be presenting it here because it will be too technical. So all these processes have been very well defined and have been established that these microorganisms contribute to abiotic stress as well as biotic stress mitigation in plant, and also enhancing nutrient status and in yield management in agriculture crops. So with this background set, I would like to share about how we are concentrating on looking at the microorganisms, or specifically on the Pseudomonas population, which are present in the finger millet rhizosphere. Actually, our research focus has been concentrating, looking into the rhizosphere region of the mangroves, as well as into agriculture crops, specifically focusing at millet. Millet was one of the crops where we looked into the diversity of the Pseudomonas alone. This rhizosphere region can have different groups of organisms. You have Azoospermillum, phosphobacteria, to name a number of organisms present in the rhizosphere, but we wanted to focus only on the Pseudomonas population, which has dual properties, which can function as a nutrient mobilizing organism as well as a disease controlling group of organisms. So, so this project, actually we initiated this project with the funding from DBT, where we looked into collected rhizosphere samples of the finger millet from different regions where finger millet is cultivated in Tamil Nadu, and we just analyzed what is the population present, which gave us an average log of 4 associated with the rhizosphere. And around, it was actually the PhD work of Dr. Jagan, who is currently with us. And with this, he was able to isolate 1,650 rhizobacteria from the root zone of the finger millet. This is just only representing only the culturable organisms. Apart from this, which represents less than 10% of the population, the remaining unculturable population are still yet to be explored. And he looked into the functions of the groups, and you find they contribute a number of plant growth promoting functions, which is shown by that Venn diagram, either by the production of ACC deaminase or AHL molecules, or by phosphate solubilization, and its salinity tolerance level, drought tolerance level, different functions. And so this gives us an idea what group of Pseudomonas, what is the population there, and what are their functions in the root zone. Next. So by using molecular techniques, we did a fingerprinting of all the isolates and grouped them, and you can see they clustered into more than 9 clusters, indicating that all these are not the same Pseudomonas group, but they belong to different strains, must be in the strain level. I won't say it is species, must be in the strain level, and the grouping is based upon different functions. And we have also looked into the antibiotic coding genes or whatever it is. Here our focus is mainly looking into the functional aspects by biochemical assay, or looking screening for the particular genes which are present in that organisms which expresses that function. Next. So, and explored into the plant growth promoting traits, where you can see in this picture, when you treat the finger millet seedlings, it enhances the seedling germination in terms of the root as well as the shoot length compared to the control, the figure at the this part shows, and the other picture, it shows how it is able to control the pathogen. In the organism in the center is the pathogen, where on either side we have streaked the Pseudomonas population, and it shows it is completely controlling the growth of that pathogen, indicating that it is a very good organism which can be used for controlling this particular pathogen. And it was also confirmed by microscopic studies and certain enzyme assays, where we can claim that this organism has a fungicidal activity and can stop the growth of this pathogen, or it can be recommended in the farmer's field for controlling blast disease in millets. Next. So again, we had done some studies with Fusarium wilt. For example, this Fusarium pathogen is a very, very hard pathogen to control because it affects the lower root zone of the plant, any plant. And here the symptom is visible only after the plant is totally affected by it. Though there are a lot of recommendations like chemicals like carbendazim and copper oxide, you find pre-treatment with these biological agent will save the plant from being affected by the wilt disease, because the spores of this fungal pathogen remain in the soil even for 30 years. You cannot destroy it with any methods you follow it. So by biological means, we have shown that the spore germination can be controlled and the spore inoculum can be brought down. And this is also supplemented by looking into the antibiotics that are produced by these organisms, and also exploring into the genes that are present, confirming that this antibiotic production happens. Next. And apart from that, after looking into the functions of the microorganisms, we look into how it enhances the plant growth in terms of seedling vigor and also its efficacy in the field, which is very, very essential for the taking up these biological inputs into the field level. Because always the problem which is faced in this research is these organisms which perform very good in the laboratory conditions does not exhibit the same properties when it is taken to the field, when it is exposed to different ecosystems or ecological functions happening in the soil. So before you promote a product which has to be adopted by the farmer, you have to ensure that its performance in the field is also well evaluated by multi-location trials. Next. And also to study the compatibility of this organism in the root zone so that it doesn't disturb the root microorganisms, other organisms which are present there. So this study shows how compatible the long streak in the center is the Pseudomonas, which we are testing, along with the other bacterial strains, which it shows that is very compatible. Suppose if it was not compatible, it will not allow the other organisms which are cross-streaked to grow, indicating it is producing certain antibiotics and inhibiting the growth of those organisms. And this was also confirmed by PFLA studies to show that this organism does not disturb the microbial population in the rhizosphere zone when you use it as a biocontrol agent or a biofertilizer. So with this research findings, we were able to have 2 good publications in high impact journal. And the next step, we wanted to test if this, which is tested individually, is compatible with other biocontrol agent and has an impact on promoting the growth in tomato also, because now the host plant is changing from the millet rhizosphere, which is a monocot. We tested it in a dicot plant along with other group of organism in a consortia mode, and we were able to show that it had enhanced activity in all the biometric parameters, indicating that this particular organism can be used even as a biocontrol agent for dicot crops also. Next. So with this background, we were able to take up major field testing through the BioFi program, which was a bilateral funding we received from DBT as well as SGC with our Swiss partners, where Dr. Madhi, who's currently here was coordinating from Swiss, and we were two components from MSSRF. We looked into the biotechnological aspect as well as the socio-economic aspect where Ranga was involved, because the focus was to test this in the field and take this technology as a package to the farmers who would adopt the use of these organisms into the for millet cultivation. And here we had extensive trial with 20 treatments and 4 replication, and the trials were conducted for 4 seasons in 2 locations, GKVK as well as Bangalore. And it had a consortium of organisms like the Pseudomonas species, AMF fungi, which was contribution from Dr. Bhagyaraj, and also Rhizobium for nitrogen fixation. So it was a combination of 3 organisms for nitrogen fixation, biocontrol activity, and phosphate solubilization. And with this trial, with this setup, the trials were conducted and we were able to come out with a good publication by consolidating these results. Then next. So just to give a brief about the findings of this trials which was conducted both in Colli Hills as well as in Bangalore. There was comparison, the impact on the use of these biofertilizer on grain yield and also straw yield was estimated by comparing the biofertilizer application to the no biofertilizer application. And always we saw when biofertilizer was applied, there was enhancement in grain yield as well as the straw yield. Next. Similarly, it was compared with the different combination with chemical fertilizers. When you just see the row below, you can see 0 RDF recommended dose of fertilizers and 50% RDF combined with the bio inputs or the 3 organisms, and only 100% RDF, where we could show that this 50% combination of the chemical input with the bio inputs yielded on par with 100% chemical inputs. And this was observed in all the 3 seasons which was tested, indicating that this can be a recommendation where you can cut down at least 25 to 30% of your chemical inputs when you use these bio inoculants as a supplement with your chemical fertilizers. So this study, So with this research findings, and we could conclude that biofertilizers contribute to nutrient enhancement and soil health management. And the next step would be to create awareness among the farmers on use of this biological input, where Ranga with her team is doing this. And we were instrumental in training these women self-help group on these production processes. Though the strain is not used for these purposes, the training happened in the microbiology lab, where the capacities were developed, and now they are able to produce this in the form of enterprise. So I just wanted to just show this slide that this technology transfer can happen if you have very good strains, and this model can be taken up, though there are a lot of thoughts, controversial thoughts, whether these eco enterprises function properly or not when compared to commercial companies. I think we have proven this as a successful model where these women are efficiently producing with appropriate trainings. So with that, I stop my research findings, and the learnings or the research questions we can derive from this is, we have looked into how this interaction helps in enhancing agriculture productivity, but looking at it from a climate change scenario, we haven't looked into its functions from the soil as a process of carbon sequestration happening or emission of greenhouse gases. So I just came across an article which was published recently in Nature, where they have said that understanding first the biological population in any ecosystem is very essential to derive models of climate change. It has to be fed into that model when you really want to look into specific climate change models, with the background saying that these organisms contribute to water retention by the production of EPS or exopolysaccharides, which is one of the functions of the organisms, and also act as plant carbon sink and carbon sequestration, and also in plant growth promotion. And the take-home message will be, with biological inputs, you can cut down on your chemical inputs for enhancing or managing the agriculture crop. And these biological inputs can be a sustainable alternate solution for crop management. Thank you. And also I record my sincere thanks to the team members and also the funding agencies who had supported this work. Thank you.
Yeah, it's very nicely presented, and perhaps hear more about the abstracts of the microbiological issues that solves the problems in agriculture. Thank you so
Thank you,
much.
sir. Thank you all.